Good morning students, how are you all? So in the last class we have started about a reproduction in plants and animals. We have seen the reproduction and its type, right? So we studied about the modes of reproduction, asexual reproduction, sexual reproduction. In asexual reproduction in animals we saw budding, binary fission, how the reproduction takes place. Yes, in plants by spore formation we have seen the mode of reproduction, fragmentation we have studied, as well as vegetative propagation. So today we will talk about natural vegetative propagation. Yes, in the last class we have studied about the natural vegetative propagation. So here, yes, it takes place by different natural vegetative propagation. We have seen propagation by leaves, propagation by roots, propagation by stems. Already we have studied this in last class. So today we will talk about advantages of natural vegetative propagation and disadvantages of natural propagation. So let us see vegetative propagation, the advantages. So here what happens, it is quick, less expensive and more certain method. So it is quick, very fast. We can, yes, the particular plant grows the vegetative propagation and less expensive. Yes, and the second, seedless plants like banana, grapes, oranges and rose can be propagated here. Yes, it is relatively cost effective and it's faster technique to obtain new varieties, you can say. So here the technique ensures the consistency of plant variety for cell. So plants do not produce viable seeds here, such as banana, sugarcane, potato. It can be raised by this technique. So here genetic characteristics of parental plants are retained and hence new varieties are not produced. So here what happens, according to the genetic character of parental plants, these are retained and new varieties are not produced. Next advantage of this natural vegetative propagation, seeds are less resistant and viable. So for example, as I said, banana, grapes, so seedless plants leads to overcrowding. Yes, it will lead to overcrowding of the plants when you start by, that is natural vegetative propagation. So these are the advantages. First one, it is quick, less expensive, is cost effective, faster technique to obtain new varieties by certain methods. Next, this technique ensures the consistency of plant variety for cell. Yes, seedless plants like banana, grapes, oranges, rose. So genetic characteristics of parental plants are retained and hence new varieties are not produced here. Seeds are less resistant and viable, leads to overcrowding. So now let us see disadvantages of natural vegetative propagation. Here what happens, risk of total destruction of an entire crop of plant, as the plants produced are prone to the same disease. So here the plant variety obtained by this technique, they lose the vigor and they become prone to the diseases. Yes, competition for nutrients and overcrowding due to fat growth. So here, yes, it will be competition for nutrients as they are growing in crowded, overcrowding due to the fat growth there. So since the offsprings are the clones identical to the parents, there is a risk of passing a deformity from parent to the offspring also increases here. So what are the disadvantages of natural vegetative propagation? The plant varieties obtained by this technique, they lose their vigor and become prone to diseases. Risk of total destruction of an entire crop of plant, as the plants produced are prone, they are identical to the parent, yes, to the same disease. Competition for nutrients and overcrowding due to what? Fat growth. Yes, so as we saw in the natural vegetative propagation, how it takes place, the advantages and disadvantages. Now let us see artificial vegetative propagation. Yes, so in artificial method of vegetative propagation, it is widely used in agriculture and horticulture. Horticulture, where the plants, the flowers, fruits, vegetables are being grown. So in artificial vegetative propagation, we are going to study four ways. The first, yes, way to perform artificial vegetative propagation is layering. So we are studying about artificial vegetative propagation. So artificial method of vegetative propagation is widely used in agriculture and horticulture. So there are majorly four ways to perform artificial vegetative propagation. So the first one is layering. So here, yes, how the particular plant is grown. You can see in the diagram, the stem of a plant is bent down until it touches the soil. Yes, here what happens? The stem of the plant is bending until it touches the soil. The stem is then cut once it develops roots. You can see, and grows into a new plant. What happens during layering? Yes, the lower branch of a plant is bent close to the ground and covered with moist soil such that the growing tip remains above the soil surface. Okay, you can see the growing tip. Yes, it remains above the tip. Yes, the growing tip remains above the soil surface. After some days, what happens? The roots can be seen growing from the branch buried in the soil. Then this is then cut and allowed to grow as a separate plant. This process is seen in jasmine flowers. Yes, so in the diagram you can see how the stem of the plant is bent down until it touches the soil, and the stem is then cut once it develops roots and grows into a new plant. Example, lemon, rose, jasmine. So the next artificial vegetative propagation is cutting. So during cutting, what happens? You can see in the diagram, leaf cutting and the stem cutting. So part of stem is cut and the cut end grows into a new plant when placed in moist soil. So when you cut a part of stem, yes, and the cut end grows into a new plant when placed in moist soil. For example, you can take mango, guava, lychee, lemon, rose. Yes, sorry, rose. So this is one artificial vegetative propagation. Next, grafting. So during grafting, what happens? Yes, you can see in the diagram, the stem of a plant is cut. Yes, or you can take twig or branch. It has been cut. It is called as scion. In the diagram, you can see. Yes, it is called as scion. Then it is detached from the plant. That is cut from the plant, and then fitted on another stalk plant, which is called as, yes, stalk. It is fitted on another strong plant called as stalk, and it is covered with grafting wax. You can see in the diagram. So here what happens? The stem of a plant is cut and then fitted on another strong plant. So the stem of a plant which is cut, it is called as scion, and it has been fitted on the another strong plant. It is called as stalk, and they are covered by what? Grafting wax. So examples we can take: apples, oranges, watermelon, ornamental plants. So the next artificial vegetative propagation, that is tissue culture. So during tissue culture is here how the particular plants are grown. It is a collection of technique used to maintain or grow plant cells. Here what happens, the tissues or organs under sterile condition on a nutrient culture medium of a known composition. Yes, for example, you take, yes, a piece of tissue is cut from a desired plant and it is kept in a nutrient medium under controlled condition, and the tissue starts growing into a mass of cells called callus. So this callus is obtained, it is placed in different nutrient medium where it starts developing plantlets. So these plantlets are again buried into pots of soil to obtain new variety of plants. So during tissue culture what happens? Yes, here the particular tissue, the piece of a tissue is cut from a desired plant and it is kept in a nutrient medium under controlled condition. Then what happens? Slowly the tissue grows into a mass of cells called as callus, and this callus so obtained is placed again in different nutrient medium where it starts developing plantlets. So these plantlets again they are buried in pots of soil to obtain new varieties of plants. So this technique is also called as micro propagation. So for example, tissue culture is, it can be used in the plants like orchid, chrysanthemum. Yes, so examples are orchids, chrysanthemum, yes, tissue culture. So next, What are the advantages and disadvantages of tissue culture? So let us talk about first advantages of tissue culture. So here in advantages, development of cell line over several generation. Yes, so the particular tissue which we are going to cut from a desired plant, yes, it will develop of a cell line over several generation. Scale up is possible. Yes, as this technique can be used to generate high yielding hybrid varieties that have special characteristics. Next, absolute control of physical environment as it has been stored in proper nutrient medium. So this technique, as I said, can be used to generate high yielding hybrid varieties that have special characteristics. Next, homogeneity of sample. Homogeneity. Homo means what? Same genes of sample. Less compound needed than in animal models. Yes, less compound needed than in animal models. Plants of desired variety can be produced through tissue culture. This is one advantage. So in advantages of tissue culture, we studied that this tissue culture experiment is performed under what? Sterile condition. Yes, because there are chances of seed-borne diseases are greatly reduced. So compared to other methods such as grafting, you take budding. This is faster and efficient method. Next, disadvantages of tissue culture. Yes, we already know that yes, many advantages of tissue culture. So disadvantages, here we can see cells may lose some differentiated characteristics. Hard to maintain also because we have to keep it in a sterile condition. Only grow small amount of tissue at high cost. De-differentiation. Yes, so there are disadvantages. De-differentiation, instability, aneuploidy. Yes, de-differentiation, instability, and yes, aneuploidy. So these are the advantages and disadvantages of tissue culture. Next we'll talk about sexual reproduction. So what is sexual reproduction? Yes, we already studied about asexual reproduction in the last class. Yes, so asexual reproduction, what happens here? Only there is involvement of one organism. Yes, it means that during asexual reproduction, only one kind of parent is involved there. Yes, there is no involvement of the sex cells or gametes. But sexual reproduction, yes, it is a type of reproduction in which male and female gametes fuse to form an offspring. So here we find two parents involvement. One is male and the other one is female. So male and female gametes, they fuse to form an offspring. So here when the male and female gametes are fusing, so the sex cells or gametes produced by males are called sperms and the sex cells formed by female gametes are called eggs or ova. So what is sexual reproduction? Sexual reproduction is a type of a reproduction in which male and female gametes fuse to form, yes, an offspring. So as the reproduction takes place, two parents are involved, male and female. So this male gamete, they produce the sex cells or gametes, you can call it as sperms, and the female gametes, they produce the sex cells called as eggs or ova, singular ovum. So next we'll talk about sexual reproduction in plants, how sexual reproduction takes place in plant. But before that, yes, let us talk about the structure of flower. So already we know that a flower is the reproductive organ of a plant. Yes, flower is the reproductive organ of a plant. So a flower may contain both the male and female reproductive organ. Yes, a flower may contain both the male and the female reproductive organ. So as I am saying, a flower is the reproductive organ of a plant. So let us understand the structure of a flower before getting to the actual process of reproduction. We see different flowers. Yes, there are of different shape, size and colors around us. but the basic structure of a flower remains the same. Yes, you can see in the diagram, as the flower stands on a stalk. Flower stands on a stalk. It is called the pedicel. It is called as the pedicel. So flower has an stalk called pedicel. The upper part of the pedicel is swollen and is called the thalamus. Yes, it is called as what? Thalamus or receptacle. So when the particular pedicel is swollen and is called thalamus or receptacle. The four whorls of a flower are supported on this receptacle. So the whorls are being supported by this thalamus or receptacle. These four whorls are discussed below. So let us see one by one. First one, calyx. So calyx, it is the outermost whorl of a flower. So in the diagram you can see in the left hand side, yes. So the long pedicel, which is called as thalamus. So on the thalamus, the four whorls of a flower are supported. So we are talking about calyx. Calyx, the outermost whorl of a flower. Calyx contains sepals that are green in color. The calyx protects the flower during the bud stage. So what is the functioning of calyx? The calyx protects the flower during the bud stage. So calyx, it is the outermost whorl of a flower. Calyx contains sepals that are green in color. Yes, so we are talking about the structure of a flower. So the flower stands on a stalk called as pedicel. Yes, when the upper part of the pedicel is swollen, it is called, yes, and it is called as the thalamus or receptacle. So this receptacle or thalamus, it will support the four whorls of flower, as you can see in the diagram. Yes, the receptacle, how it is supporting the four whorls. So the first part, let us talk about the calyx. So calyx, you can see in the diagram left hand side corner, is green colored at the center. Yes, the calyx, it is the outermost whorl of a flower. Calyx contains what? Sepals that are green in color. The calyx protects the flower during the bud stage. Next, corolla. Yes, corolla. So after the calyx, as the calyx is the outermost, corolla, it is the second whorl of a flower and is divided into segments. You can see there 3, 4, 5 segments. Yes, corolla, it is the second whorl of a flower and is divided into segments. Each segment is called petal. So each segment is called as petal. The petals are brightly colored because of the presence of pigments. Yes, petals are brightly colored. You can see in different flowers different colored petals. So it is due to the presence of pigments present in it. The sweet smell, bright colors of the petals and the nectar attract insect to a flower and help in pollination. So the sweet smell, yes, of the petals, it will attract the insects. The bright colors of the petals as well attracts the insects. Yes, and this helps in the pollination. Yes, students, we'll stop up to here, and the next part, that is stamen or androecium, pistil or gynoecium, we'll take it in the next class. Yes, so what we studied today about advantages and disadvantages of natural vegetative propagation. Yes, as we saw in vegetative propagation, it is quick, less expensive and more certain method. Seedless plants like banana, grapes, oranges and rose can be propagated. Genetic characteristics of parental plants are retained and hence new varieties are not produced here. Seeds are less resistant and viable. It leads to overcrowding. Next we saw about disadvantages of natural vegetative propagation. Here the plant varieties obtained by this technique lose their vigor and become prone to diseases. A risk of total destruction of an entire crop of plant as a plant produced are prone to the same disease. Competition for nutrients and overcrowding due to fat growth. Next we saw the artificial vegetative propagation, which is widely used in agriculture and horticulture. So which are the four ways to perform artificial vegetative propagation? First one, layering. So layering examples we can take lemon, rose, jasmine. So in layering what happens? The lower branch of a plant is bent close to the ground and it is covered with moist soil such that the growing tip remains above the soil surface, and after some days you can see the roots can be seen growing from the branch buried in the soil. So this is then cut and allowed to grow as a separate plant. Layering. Here the stem of a plant is bent down until it touches the soil. The stem is then cut once it develops roots and grows into a new plant. Next we saw cutting. Here what happens? A part of stem is cut. Is commonly observed in rose, sugarcane. In this process, a healthy young branch that has nodes, internodes is selected. The branch is then cut and its leaves are removed. Then the branch is buried in the moist soil. After a few days, roots can be seen coming out of the buried branch. This eventually develops into a new plant. Next we saw about grafting. Here what happens? The twig or branch, the stem of a plant is cut. It is called a scion and it is fitted on another strong plant called a stalk and covered with grafting wax. Example you can take apples, oranges, watermelon, ornamental plants. Then next artificial vegetative propagation we saw about tissue culture. A collection of technique used to maintain or grow plants. Here what happens here? The tissue, a piece of tissue is cut from a desired plant and is kept in a nutrient medium under controlled condition. Slowly the tissue starts growing into a mass of cells called callus. The callus is obtained, is placed in different nutrient medium again where it starts developing into plantlets. These plantlets are buried in pots of soil to obtain new varieties of plants. This technique is also called micropropagation. So tissue culture is used to propagate in orchids as well as chrysanthemum. Next, what are the advantages and disadvantages of tissue culture? Advantages we can see there is development of cell line over several generations. Here, as the plants of desired variety can be produced through tissue culture. Compared to other methods such as grafting, budding, this is faster and efficient method. Scale up is possible, absolute control of physical environment, homogeneity of sample, less compound needed than in animal models. As well as this is performed under sterile condition, the chances of seed-borne disease are greatly reduced. Next disadvantages we studied about of tissue culture. Cells may lose some differentiated characteristics. It is also hard to maintain. Only grow small amount of tissue at high cost. As well as dedifferentiation, instability, aneuploidy. Then we spoke about sexual reproduction. So what is sexual reproduction here? It is a type of reproduction where two parents are involving. One is male and the another one is female gamete. So the male gamete and the female gamete, they fuse to form an offspring. Because the sex cells or gametes produced by males are called sperms and the sex cells formed by female gametes are called as eggs or ova. The fusion of sperm and eggs, it is called as sexual reproduction. Ovum singular. So sexual reproduction in plants. As we all know, flower is the reproductive organ of a plant. So there are different varieties of flowers we can shape. We can see the flowers of all different shapes, sizes, colors around us. So flower may contain both the male and female reproductive organ. So let us study about the structure of flowers. In the diagram you can see Yes, flower stands on a stalk called the pedicel. The upper part of the pedicel is swollen and is called as what? Thalamus. Yes, or you can see it as receptacle. You can see in the diagram. Yes, receptacle. So the four whorls of a flower is supported on this receptacle. These four whorls are discussed below. So we have studied about two whorls, that is calyx and corolla. So the first one, calyx, the outermost whorl of a flower. Calyx contains sepals that are green in color. You can see in the left hand side in the diagram, green colored. Yes, calyx contains sepals that are green in color. So calyx protects the flower during the bud stage. So calyx, what it does, it protects the flower during the bud stage. Next, we studied about the next whorl, that is corolla. Yes. So after the calyx, as calyx is the outermost, corolla is the second whorl of a flower, and it is colored one. In different flowers you can see different colored corolla which are divided into segments. So each segment it is called as petal. So petals are brightly colored because of the presence of pigments. We can see in different flowers different colored petals. So these different colored petals, they are sweet smell, bright colors of the petals, they attract the insect, and the nectar attract insects to a flower and help in pollination. Yes, students. So in the next class we will be studying about the next two words, that is stamen and pistil. So today we will stop up to here. If you have any doubts, we will clear it during our offline class. Okay, thank you.